Let's begin DNA replication review, beginning with prokaryotic DNA. Prokaryotic DNA is circular and it only has origins of replication. This makes things simple. If you remember, the origin of replication is simply a sequence or region in the DNA where replication begins. Enzymes that carry out replication initially bind to these sites. As you can see, a replication bubble is forming, as well as two replication forks moving in opposite directions. Soon, you will have two daughter molecules of prokaryotic DNA. This is fairly simple and intuitive when compared to the complex process of eukaryotic DNA replication. First, we have our enzyme of topoisomerase. The job of topoisomerase is to relieve tension in the DNA molecule. Remember that the DNA molecule is being unraveled. This creates a large amount of stress. Topoisomerase breaks apart DNA and rejoins it in front of the replication bubble. Next, we have helicase. Helicase unravels and helps to separate the parental strands. This is furthered by the single strand binding proteins, which make sure that the two parental strands do not rejoin back together. Now we have our RNA primers. Remember that DNA replication is a heavily enzyme catalyzed reaction. However, this is not to say that enzymes can initiate replication. We need RNA primers to show where we want the replication of our DNA to begin. This is usually a very short nucleotide sequence that matches with the parental strand or is complementary to it. Here we have our leading strand. Remember that DNA only goes from 5' to 3' when it replicates. There is no problem with this strand because we're going from 5' to 3' and we can go continuously. As we go from the 5' to 3' direction, DNA polymerase 3 fills the replicated strand with complementary DNA. A problem arises with the lagging strand. When we go from the 5' to 3' direction, our RNA primer is from the 3' to 5' direction, but we can only build DNA from 5' to 3'. We solve this by creating another RNA primer going from the 5' to 3' direction. Thus, we can now go from 5' to 3' in the middle of these two primers. That is the job of DNA polymerase 3. Afterwards, DNA polymerase 1 removes the RNA primers and replaces them with DNA. DNA ligase then ligases together these individual fragments so that they form one continuous strand. This is an overview of the process. There is a replication bubble being formed with two replication forks moving in opposite directions. The dark dots that I've drawn are the origin of replication. Going from this origin of replication to the 5', we are going from 5' to 3'. There is no problem with this. Going from this origin of replication to the 3', we are going 3' to 5'. There's a problem here, and so this is our lagging strand. We call each of these fragments in the lagging strand Okazaki fragments. There's an interesting phenomenon that occurs during DNA replication, especially at the ends. Remember that in the lagging strand, we are using multiple RNA primers. Here I've depicted our RNA primers. DNA polymerase 3 then moves in and fills the gaps with DNA. Let's do that. Imagine now that the DNA polymerase 1 has also come in and removed these primers and replaced them with DNA. The problem now, as you can see, is we have a gap. So what do we do about this gap? Well, in theory, every time we replicate, we're losing DNA. Eventually, we will lose all our genetic material. But luckily, our body has a way to prevent this. Remember that telomeres are at the ends of our DNA. They're involved with DNA replication and the maintenance of chromosomal ends. There's actually a way to elongate our telomeres by a telomerase enzyme. Telomerase is a special enzyme that maintains the structure of telomeres. It is a ribonucleic protein, a complex of protein and RNA that contains an RNA template for the telomeric repeat. The telomerase enzyme contains an RNA template that allows the template strand to be lengthened by the addition of more telomere repeats. It contains code for a new telomere repeat, plus a little bit of an overlap to make sure that there is proper positioning. In this case, we might have this code as our telomerase enzyme. Notice that there is a bit of an overlap. Now, the code is simply copied over. Now, we remove the telomerase. With the extension, RNA primers can now attach here, and DNA polymerase 3 can fill that with DNA. Now we're missing this part, but remember that we elongated our telomere ends, so that part doesn't really matter. It's not going to be coding for anything important. And that's an overview of DNA replication.